Hello, everyone, and welcome to today's webinar, which we are pleased to host in recognition of April being Parkinson's Awareness Month. Research Challenges in Early Parkinson's Disease, Diagnosis, Trial Design, and Rater Training. My name is Colleen Lezovitz, and I'm a Senior Marketing Manager at Worldwide Clinical Trials. Before we kick things off and dive into the presentation, I did want to go over a few things. All attendees are on mute, but please use the Q&A window to submit questions at any time pertaining to the presentation, because we will be reserving time to answer your questions at the end of the session. This webinar is being recorded, and all attendees will receive a link to view the on-demand, so be on the lookout for an email, most likely tomorrow. At this time, I would like to hand things over to Dr. Babich. Dr. Babich?
Thank you, Colin, very much. Good afternoon, good morning, everyone. This year we are celebrating 200 years since British neurologist James Parkinson published Essay of Shaking Palsy, where he described 6 people with a new, completely different clinical picture with a tremor and rigidity. And a few years later, thankfully to Charcot, this disease was called Parkinson's disease. After 200 years, the knowledge of disease, biochemical knowledge, biomarker knowledge, clinical knowledge, substantially progressed. And today we are still, of course, having plenty of unanswered questions regarding Parkinson's disease, but there are plenty of important clinical facts that we are aware of, and we can probably have a good opportunity to treat our patients. As you can see, the prevalence and incidence of Parkinson's disease is age-dependent. This is very much similar to Alzheimer's disease or to stroke, which are both belonging in some extent to neurodegenerative disorders. And there is no specific difference in terms of geographic regions regarding idiopathic Parkinson's disease. Of course, there are some type of Parkinsonism, which is Parkinson's syndrome, which is more common in some countries, usually related to pesticides or to poisoning with some special substances like manganese in China or something like that. But anyway, we can conclude that Parkinson's disease is in fact age-related disease with the usual occurrence in the early 40s, and 5 to 10% of all patients are belonging to so-called genetic type of Parkinson's disease, which will not be today's topic. We will today discuss early idiopathic Parkinson's disease, challenges about diagnosis of early Parkinson's disease, and how clinical trials will be done. So Parkinson's disease is a progressive disorder, phenotypically characterized by akinesia, resting tremor, and muscular rigidity. And this classical clinical expression is a consequence of complex pathophysiological processes in substantia nigra and also other basal ganglia in the brain, leading to intraneural accumulation of protein called alpha-synuclein, which is in fact forming a Lewy body. It seems that the spreading of alpha-synuclein goes from neuron to neuron, and alpha-synuclein accumulation, as you will see later, is not specific only of brain. There is alpha-synuclein in other parts of our body. It seems that nearly 60% of dopaminergic neurons should be destroyed by alpha-synuclein and mitochondrial dysfunctions to have a clinical, motor clinical syndrome available, where bradykinesia looks as the symptom which is best correlated with degrees of dopaminergic neurons loss. What is also important as well is that there is something like a magic circle in which alpha-synuclein aggregation and mitochondrial dysfunction exacerbate each other, which can be explained why these cellular changes are observed together in degenerating neurons in Parkinson's disease. As a consequence of this oxidative stress, disruption of calcium homeostasis, abnormal kinase activity, we have neuroinflammation with activating microglia as one of the vital degenerative role among dopaminergic neurons. So this is really a complex disorder, not only CNS related, that you will probably see in the next few slides. The classical expression, as I said, is bradykinesia, rigidity, and tremor, as you can see. But in fact, during the early accumulation of alpha-synuclein, usually in brain stem, and spreading of alpha-synuclein towards cortex and basal ganglia, there are some non-specific, non-motor symptoms which might be potential clinical biomarkers for patients with Parkinson's disease. Mainly, this is loss of smell sense, constipation, depression, usually articular pain, fatigue, and orthostatic hypotension. Something which is not entered here in this picture is a symptom called rapid eye movement sleep behavior disorders, which is frequently present in Parkinson's disease. And it seems that the presence of this symptom is very important for later development of Parkinson's disease. And we have now data that practically among 80% of patients with initially present with rapid eye movement sleep disorder will develop either Parkinson's disease or Lewy body dementia, which are both alpha-synucleinopathies, where we have accumulation of alpha-synuclein in our brain. This latency, when first occurrence of the rapid eye movement till motor symptoms present of Parkinson's disease, can be between 12 and 14 years long. So apparently the evolution of disease starts much long time before we have a classical clinical expression as a tremor, rigidity, and bradykinesia. So diagnosis of early Parkinson's disease is it's sometimes quite difficult, because every physician has his own impression of how Parkinson's disease looks like. And unfortunately, in everyday clinical practice, very rarely you will find physicians who are using criteria for Parkinson's disease, because criteria for Parkinson's disease usually have been developed for clinical research. So you can see here 4 cardinal symptoms of manifested Parkinson's disease. Tremor at rest as probably the most common symptom. Bradykinesia, most important, which will definitely be present for diagnosis. Rigidity of muscles, and impairment of postural reflexes. In the new diagnostic criteria, impairment of postural reflexes are not something that is mandatory for diagnosis of so-called Parkinsonism. So in fact, when we are talking about Parkinson's disease, we basically have a tremor at rest, bradykinesia, and rigidity as symptoms and signs of Parkinsonism, which is Parkinsonian syndrome, which might be present in various conditions, not only in idiopathic Parkinson's disease. However, the diagnosis of Parkinson's disease, according to some criteria, should be done with the exclusion of some conditions, for instance, mass lesions in the brain, or also some lesions of a pyramidal system. And also there are some supportive symptoms and signs which we are using, for instance, unilateral presentation of symptoms at the beginning, when Parkinson's disease, idiopathic Parkinson's disease, should be diagnosed. For clinical trial purposes, we have two quite similar, but also different criteria for diagnosis. The most common and the older one is UK Brain Bank diagnostic criteria, And the new one, recently published 2 years ago, is a Movement Disorder Society diagnostic criteria for Parkinson disease. What is important for diagnosis of Parkinson disease in early stage is, in fact, presence of various biomarkers. We need to point out that these biomarkers are not sufficiently sensitive and sufficiently specific to be sufficient for diagnosis of Parkinson disease if they are present alone. But a cluster of biomarkers together with a clinical picture is very important and very powerful tool for diagnosis of Parkinson disease. If we skip genetic biomarkers, which are definitely related to genetic a genetic type of a Parkinson disease, we can only say that there are some genes which are important in Parkinson disease. So, for instance, we know today that heterozygote people with a glucocerebrosidase enzyme, which is in fact important for Gaucher disease, these people can have a Parkinson disease more commonly than others. And we know today that particularly among Ashkenazi Jewish, there are 10% of patients with Parkinson disease who have a positive glucocerebrosidase homozygous mutation for this gene. More important are biochemical biomarkers, particularly because plenty of these biomarkers are using in clinical trials. This is usually the most common and most important is alpha-synuclein protein in CSF, but it's important to say that alpha-synuclein can be found in erythrocytes, thrombocytes, can be found also in the gut mucosa, colon mucosa. So in fact, alpha-synuclein in the CSF usually is very difficult to explain, because if you have artificial blood during the lumbar puncture, which is very common, practically you have a contamination with alpha-synuclein, which is coming from erythrocytes. That's why it's very important to have a minimal erythrocytes in a CSF if you are assessing alpha-synuclein. At the moment, we can conclude that alpha-synuclein in CSF is not good marker, neither for diagnosis nor for longitudinal assessment of a drug effect in patients with a Parkinson disease. Alpha-synuclein can be found in saliva as well, and also, as I already said, we found this in the colon and also in the skin. More important biomarkers at the moment are neuroimaging biomarkers, where dopaminergic PET scan is sufficiently sensitive in identifying dopamine deficiency even during the preclinical disease, and it is potentially useful in quantifying disease progression. However, there are a number of challenges associated with PET scan. Usually, interpretation of results may be affected by compensatory changes resulting from disease and pharmacological intervention. And dopaminergic PET is quite expensive, needs specialized infrastructure and expert analysis. Recently developed data scan, which is eofluorescence single photon emission tomography, is a widely available and less expensive tool, which is already approved for routine practice. It is also serving as a help in diagnosis, but it's not good enough for longitudinal assessment of the drug efficacy. And also there is a classic FDG PET, which is PET assessing glucose utilization. It is helpful in differential diagnosis of Parkinsonism in early stage, and also results should be affected by compensatory mechanism. Structural MRI is important at the moment to rule out other potential disorders, and there are new MRI techniques which with some promise in detection of Parkinson disease quite early, which is MRI assessing iron accumulation in substantial nigra during the Parkinson's disease progression, but this is still not in a clinical usage or not in a clinical trial. And finally, transcranial ultrasound is helpful in early stage of Parkinson's disease. It is cost-effective, show promise as a possible imaging biomarker, but it's very dependent on rater operator skills, and also the adequate temporal acoustic bone window is needed for a good imaging. Apart of all of these dopaminergic biomarkers, there are a few non-dopaminergic biomarkers useful in Parkinson's disease, and the most important is the in fact biomarker who is assessing cardiac sympathetic innervation, and this can be seen in patients with Parkinson's disease as a decreased uptake of the sympathetic marker, and this is a so-called 1,2,3 meta-iodobenzylguanidine (MIBG), using as a cardiac SPECT. This biomarker is quite specific for Parkinson's disease. It serves for differential diagnosis between Parkinson's disease, Lewy body dementia, and multisystem atrophy. And also, what is very important, this is the only one biomarker which was included as a supportive tool in a newly published diagnostic criteria from Movement Disorder Society groups. Also, there are some neuroinflammatory biomarkers which are assessing microglia, which is a PBR028, using by PET, and we investigated some patients using this particular biomarker. Why this is important for clinical trials, and what is in fact the role of these biomarkers in clinical trials? So you can see here that, as we've seen, neurons are basically becoming less functional, and also deterioration and progression of neuron destroying is present in patients with Parkinson's disease plenty of years before motor symptoms and signs. And during this premotor period, we basically have some factors which are important in patients with Parkinson's disease, and also some premotor markers which we can use as a potential diagnosis of prodromal Parkinson's disease, where we can plan clinical trials of the disease modification as a primary prevention. The primary outcome in these clinical trials should be time of the diagnosis, or time of the certain UPDRS change in motor symptoms and signs. And unfortunately, at the moment, there is no clinical trial in prodromal Parkinson's disease, but definitely this is one of the most promising area in terms of the research of disease modifying treatment. At the moment, there is no drug in clinical research or drug which was investigated to be sufficiently efficacious for disease modification in this stage. And finally, there is another group of early Parkinson's disease clinical trials where we have already motor symptoms present. This is usually early Parkinson's disease in clinical settings, where we have bradykinesia, rigidity, and tremor. And in this stage, we can plan two different types of clinical trials, which one is a symptomatic treatment with a new compound with or without levodopa, and the second one is prevention of a levodopa-induced complication, because we know that levodopa definitely will lead to complications during the treatment. So in this stage, there are two different types of trials which might be implemented. However, it's important to say that in this stage, patients have a good option for the standard treatment, which is either levodopa or dopaminergic agonist. So basically, that's why recruitment rate in this patient population is much lower than in advanced Parkinson's disease, which will be addressed by talk of my colleague Trevor today. So anyway, I would stop now, and I would hand over to my colleague Tamara asked. She is a vice president of project management, and she will be talking about operational aspects of a Parkinson disease. I'm sorry, I have a problem how to generate next slide.
Okay, I think it is fine now. Thank you very much, Tom. So in this next section, we will cover some of the key operational considerations related to trial conduct. As I'm sure you will all agree, appropriate site selection is of the utmost importance, especially for the early phase studies, for phase 1 and phase 2 trials that will not have a large number of sites. We need to ensure that every single site can contribute the planned number of patients within the agreed timelines. On a small study with 10 to 15 sites, 1 or 2 sites not performing can really have a detrimental effect to the recruitment timelines. So with this in mind, we always aim to have the right balance between key opinion leaders and well-recruiting investigators. Feasibility assessment is an important step that's required to confirm the investigator motivation for the particular trial we are discussing, and also to check the current status of competitive studies, both per country and at the site level. It is a good approach to explore some contingency sites or contingency countries, and our recommendation would always be that these are pre-qualified and ready to activate in case required. It is too late to start looking for backup sites or additional sites halfway through the study once we are experiencing some challenges with recruitment. Another key consideration for Parkinson's disease trials is the importance of caregivers. They will be key both in patient recruitment as well as in ensuring good patient retention. And it is also important to understand the different approach that is required when discussing the study with caregivers who are adult children as opposed to caregivers who are elderly spouses. As Tom has already indicated in his presentation, the protocol should be scientifically well designed, but also kept simple and doable. So we would strongly recommend passing on the final draft of the protocol to operational team for their input and review prior to finalization. We have often found that statisticians recommend setting up the upper limit of randomized patients per site to avoid any site-level bias. And finally, I'd like to stress the importance of appropriate investigator grant fees. These can have a significant impact on recruitment, especially at sites that have other ongoing trials at the same time. Rater training is another key operational consideration, and my colleague Rolana will be covering this topic in greater detail shortly. Moving on to patient recruitment strategies. It is important to be aware of different standards of care in different countries in case you are planning a global trial. We have found in our recent trials that the use of non-traditional medication, herbal supplements, tends to be higher in some regions, for example in Eastern Europe, especially with early Parkinson's disease patients before the formal diagnosis is confirmed. In case some concomitant or rescue medication will be mandatory during the course of the trial, again, it is important to check during the early stages of the trial setup that these are available in a given country or to make appropriate plans for importation. We should also make every effort to ensure that we are providing adequate support related to patient participation. Providing support with travel logistics or reimbursement can definitely enhance recruitment. If the order of study assessments is important, this should be clearly outlined in the protocol so that it is cleared for all the site staff what is the order of procedures to be followed. And these should be planned in such a way to ensure the most efficient use of patients' time during the on-site visit. In some of the recent trials, we have found that having 6 hours on site for the patient for studies that require intensive PK sampling can be, very tiring. So we should make every effort to minimize the amount of these very lengthy visits. When it comes to the individual sites recruitment, our experience has consistently demonstrated that targeting sites that have their own database of patients or a strong GP referral network, this is especially relevant for early PD patients, this will produce, far better results. Consenting process is one of the key challenges in early Parkinson's disease, and we find it that patients are far more likely to be interested to take part in a clinical trial if they have an existing relationship with the investigator and if they know that the same doctor will continue to look after them even after the trial completion. As already mentioned, appropriate caregiver approach will significantly enhance patient recruitment. And with this in mind, we would recommend preparing study information leaflets that are specifically aimed for caregivers. Finally, sharing experiences and success stories is a great way of keeping investigators engaged and motivated. We would recommend keeping the study teams up to date with regular newsletters and potentially some joint investigator calls. Another approach that is always very welcomed by the sites is face-to-face sponsor site visits. We find it that the sites very much welcome the opportunity to discuss the study directly with the sponsor, to provide you with their feedback and hear directly from the sponsor about future development plans for the drug that is being tested. Once the study sites are activated and ready to start recruiting patients, one of our main goals is to reduce the amount of screen failures and at the same time ensure high retention rates. This is achieved with good investigator training during the investigator meeting and also during the site initiation visits. As you can see on this slide, there are many predictable factors that contribute to screen failures, and our goal is to ensure that all of these are addressed prior to patient even being consented for the study. The investigator and the study team should be fully familiar with the protocol, and if this is the case, we should not see any screen failures, for example, due to medical history or concomitant medication or logistical issues. Once the patient has been screened, there are, of course, a number of non-predictable factors that can result in screen failures, like MRI data, lab data, ECGs, and so on. However, there are also some semi-predictable aspects that we can address during a consenting process and ensure that patient and, if appropriate, caregiver are fully aware of what the study burden will look like and what is the time commitment required during the course of the study. Following the subject randomization, we will inevitably lose some patients due to other non-predictable factors like adverse events coming up. But again, we should make every effort not to have dropouts due to predictable factors like lack of benefit, length of the study, dosing frequency, and so on. Oopsie, I went too much. So in summary, in order to meet the study objectives within timelines provided, we should really strive to have a state-of-the-art protocol that clearly defines the study population, endpoints, and assessment tools. Investigator selection is key. We are looking for highly motivated sites with proven quality of performance. Study management team should be comprised of therapeutically aligned team members with a very proactive approach. And finally, we should come up with realistic timelines, neither too short nor too long. And my colleague Travis will touch upon this topic in the next section of the presentation. Over to you, Travis.
Thank you very much, Tamara. All right, I'll be talking today a little bit about the enrollment characteristics of these early stage Parkinson's disease trials. But as my colleague Dr. Babich alluded to, this really begins with understanding patient availability. You know, one of the many tools we have to look at patient availability are EMR systems, and a particular system we accessed in advance of today's webinar looks at 50 million US lives, and within these 50 million US lives, there were 174,000 patients with a diagnosis of Parkinson's disease. And the blue folks on the left of the slide, they represent those 174,000 patients. But when we begin applying common inclusion-exclusion criteria for early stage Parkinson's disease trials, we can see how quickly that field of potential patients narrows. When we look only for recently diagnosed patients, those who were diagnosed in the previous 12 months, you know, our sample size immediately falls to around 9% of that 174,000. When we look at that 9% and we specifically target those who've not had exposure to levodopa or dopamine agonist, you know, now we're looking at around 5% of that total number of patients. And really, we have many other inclusion-exclusion criteria that are going to whittle down that 5%. So when we talk about enrolling and the enrollment rates we're going to achieve in these early stage studies, you know, we're really talking about studies that are enrolling less than 5% of the available Parkinson's disease patients. And again, this 50 million is somewhat representative of the US market, but we expect to see similar patterns in other countries as well. When we look at competition in terms of competing clinical trials, we know that this dramatically impacts the enrollment potential of trials. You'll see on the left Parkinson's disease trial starts. The orange bars represent all Parkinson's disease trial starts, and the blue bar represents just early stage Parkinson's disease trial starts. And you can see that we've had some fluctuation. The number of trial starts for early stage Parkinson's disease did peak in 2011 and then receded for a couple of years. But as we've seen the number of total Parkinson's disease trials begin to climb in the last 2 years, we expect the number of early stage trials to climb as well. Although this looks at trial starts and not trial duration, when we look at the current competitive situation globally, you can see that the countries with the greatest amount of competitive activity in North America and Western Europe have up to 12 early stage Parkinson's disease trials running concurrently. And again, that's 12 studies, you know, competing for around or under 5% of potential Parkinson's disease patients. When we look at the availability of investigators, you know, we see that a predominance of the work done in this area has been done in North America and in Europe, specifically Western Europe and Central Eastern Europe, although we do see involvement in other regions in Asia and in Latin America. When we think about some common questions with respect to early stage Parkinson's disease trials, you know, we did an analysis of of all early stage Parkinson's disease trials that have occurred over the last 10 years or so, and looking across that trial and answering the question, how long do early stage Parkinson's disease trials typically take to enroll, we were able to use a box whisker plot analysis here, and can answer that question that most enroll in 8 to 20 months. Now that's a pretty wide range. The 8 represents the observed 25th percentile, and the 20 months represents the observed 75th percentile. The median enrollment for early stage Parkinson's disease trials is around 12 months. When we think about how many sites are typically involved, obviously there's going to be a correlation between the number of sites involved, the number of subjects, and the enrollment time. But generally, again, applying the same box whisker plot methodology, we see that most include between 7 and 50 sites, with around 25 being the median number of sites included. And then finally, these studies are enrolling typically enrolling between 60 to 300 subjects, with 175 representing the median value. So we get a sense that most studies are enrolling around 12 months, with around 25 sites, and around 150 to 175 subjects. From an enrollment rate perspective, it's very difficult to be able to survey the entire landscape. We've pulled a selection of trials here where the actual enrollment data in these trials is established within the public domain through publication. But you can see, as my colleague Dr. Babich indicated, the enrollment is very modest, especially compared to late-stage Parkinson's, where more aggressive enrollment rates can be achieved. I'm now going to hand it over to my colleague Rolana, who will speak about training and eligibility reviews.
Great, thank you, Travis. Yes, today I'll be speaking with you about the training considerations as it applies to the UPDRS, and also, of course, to other key measures in Parkinson's studies, as well as the importance of eligibility reviews and associated data surveillance quality methods. Let's see if I can advance. Okay, so what we, of course, are aiming for is a successful clinical trial, and that is largely dependent, of course, on having reliable, high-quality data. So how do we achieve this high-quality data? Well, there are a number of factors that come into play, and that includes the scales chosen for the trial, knowing what is being assessed, identification of the raters in the trial, particular rating challenges that can come up, as well as planning for an appropriate rater training program and eligibility review and surveillance program to mitigate any risks that are associated with the challenges. So as many of you are probably quite familiar with, the UPDRS is a very common scale used in PD trials, as well as the Hen and Yahr and ADL scales, such as the Schwab and England ADL scales. These measures will look at non-motor and motor experiences of daily living, dyskinesia, dystonia, on-off periods, and functioning. Now, specifically to on-off periods, this does not apply to early PD trials, but more towards the advanced PD trials. But the UPDRS will cover all of these areas. In particular, for early PD, we are interested in part 3, which is the motor examination of the patient, and the rater will then look at various considerations such as speech, facial expression, rigidity, finger tapping, freezing of gait, and so on. Parts 1 and 2 of the UPDRS rely on patient and or caregiver information regarding their functioning. So who are the raters in a trial? Well, as I mentioned, you have your clinic rater, your patient rater, and your caregiver rater. And there are a number of challenges to consider with regard to ratings. Because you have clinic, patient, and caregiver raters, there are multiple sources of information. And this can lead to variability in the data that's coming into the trial. There are varied levels of experience, which can impact the accuracy of the assessments of symptom severity. Also need to take into account caregiver impact, issues related to placebo response, and overall consistency. So in order to mitigate these risks associated with these challenges, a comprehensive training program should be put into place. And this training should be provided to all study raters, not just the site raters, but also patient and caregivers as well. As my colleague Tamara noted, that information regarding the study should be provided to caregivers and patients, and this should also include scale information and expectations of the trial as well. Rater training allows for consistent approaches to ratings from the very beginning. This is aimed to reduce variability in the data and to ensure a standardized approach to ratings. And it should focus on scale and protocol specifics as well. Following a rater training program, it is highly advisable to institute an eligibility review process and clinical data monitoring. This would address issues of potential rater drift, but will also ensure appropriate patient inclusion into the study. So as I noted, there are particular challenges involved with rating in the UPDRS. In particular for the clinic rater, we need to take into account their experience with the indication itself as well as the scale. For Part 3 of the UPDRS, the rater must accurately evaluate rigidity in multiple areas of the body. And when determining ratings that are slight, mild, or moderate, there can be a lot of variability in how a rater approaches these ratings. As my colleague Dr. Babbage noted, experienced raters or investigators will have their own experience to draw from, their own perceptions of what a symptom would look like and how a symptom expresses itself. So how one would perceive a slight severity can differ from another. Other challenges to consider with patient and caregiver raters as they are providing information regarding their functioning and their behaviors and emotions. There are expectations that they may have of the trial itself, of the treatment, of the site that can influence their responses to rating scales. There are emotions that can come into play, particularly for the caregiver as well, taking into account caregiver involvement and potential caregiver burden. There's a notion that heightened emotional responses or negative emotions can contribute to a negative feedback loop of worsening symptoms, and this can color their perception of symptom severity and overall functioning. So this can influence their ratings on key measures. It is important to consider consistency. There should be consistency across all raters, the site and the caregiver in particular. The caregiver should be very familiar with the patient. All best efforts should be made for the same site rater to follow the patient because they become familiar with the patient's symptoms, and can more accurately evaluate the worsening of symptoms from visit to visit. Of course, there will be times where the same site rater may not be available, but again, best efforts should be made to maintain this consistency because different raters can introduce variability in data. As I noted earlier, placebo response is a consideration as well, so there should be a focus on the relationships between the site and the patient and the site and the caregiver, and an understanding of what the expectations are of the site and of the study. An overall comprehensive training program should address all of these concerns. So with regard to training considerations, what we should start off with is a clear and thorough look at rater experience. Do an assessment of the raters who are being proposed for the study. This includes an overall review of experience in the indication, as well as experience with outcome measures and experience with UPDRS. One can have, for instance, 20 years clinical experience, very strong clinical experience, but have limited experience with the UPDRS. And so it is important to get an overall picture of the site raters who are coming into the study. Rater training should be done by trained and experienced presenters, oftentimes by KOLs. Didactic training should be focused on the specifics of the scale and also protocol specifics, so the raters are aware of how the scale is to be used in the study. But the scale information should be thorough, should go through each item, scoring anchors to give a clear understanding of what is expected from the raters. And while webinars are, of course, highly effective and useful, face-to-face training such as IMs are highly recommended. This would allow for a more interactive approach, allow for more discussion between the trainer and the site raters as well. Videos are of great help in trials that utilize the UPDRS. They should cover administration scoring guidelines, but most importantly, they provide a clear visual description of symptom severity. So the raters viewing the video will have a clear example of various issues regarding gait arising from chair, tremors, what different severity levels look like, and so forth. And oftentimes it would be advisable to include a scoring option to go along with the video, so the raters can score along and compare the scores to gold standard ratings to ensure consistency and calibration across the site raters. Mock interviews are highly effective as well at IMs. This is where you would utilize breakout sessions, and this is where a site rater would then work with a standardized patient or a mock patient in a room observed by an experienced clinical observer. They will then do a short interview or do an assessment, particularly Part 3 of the UPDRS. The clinical reviewer will assess for accuracy in terms of interviewing and scoring, and should provide real-time feedback to realign the rater with the administration scoring guidelines. It is an opportunity for the rater to apply the knowledge that's gleaned from the didactic training into practice. And again, placebo response is, of course, an issue, and so it should focus on site interactions, both with the patient and the caregiver, give full information regarding caregiver impact, and provide training on not just what the patients and the caregivers should expect as far as their interactions go with the site, but also information regarding the study and the scales themselves. And following a thorough training, eligibility reviews are highly recommended, and this can be done through source monitoring or even video monitoring, where an independent experienced clinician should review source notes or videos of the actual interview themselves to ensure that interviews are being administered correctly, that the scoring is correctly, and they can provide real-time feedback to the raters to realign them with the conventions and to address issues with potential rater drift. This will also ensure appropriate patient inclusion. Beyond the eligibility review, it is also important to utilize these types of monitoring programs along with potential data surveillance program to identify, again, any rater drift, any inconsistencies concerning clinical inconsistencies between scales and visits in an effort to decrease variability and increase inter-rater reliability. And With that, I will then turn this over back to Colleen to start our Q&A section.
Thank you, Rolana. At this time, we invite all attendees to use the questions window pane to submit any questions you have for our speakers. So I'll give everybody a little bit of time to submit their questions. Okay, it looks like we have some coming in. So the first question, what is the clinical difference between idiopathic Parkinson's disease and Parkinson plus syndrome in the early stage of the disease?
Well, this is Tom. This is a very good question. In early stage of Parkinson's disease, we know even if we are strictly using criteria for clinical research, there is about 20% of patients who are expressing with a clinical picture of Parkinson's disease, but in fact they have something else, either multisystem atrophy or progressive supranuclear palsy or dementia of Lewy body. So in fact, one of the most important parts of the clinical picture in early stage is asymmetry. Asymmetry is a feature of Parkinson's disease, so practically all of these Parkinson plus syndromes, including Parkinson's disease related to drug abuse, particularly neuroleptics or treatment with neuroleptics. So all of them have a symmetrical expression either of bradykinesia, rigidity, or tremor if it is present. So asymmetry is something which is a leading feature of idiopathic Parkinson's disease. In addition, tremor is very rare in Parkinson plus at the beginning, and tremor at the rest is usually first symptom of idiopathic Parkinson's disease. However, we can always be in two or more minds regarding the differential diagnosis. So in that sense, we might use some biomarkers, as we already said. One of these biomarkers is data scan. So in fact, we can see also asymmetry in basal ganglia in terms of destroyed dopaminergic neurons, and similarly, cardiac sympathetic SPECT also will be of a great help to distinguish idiopathic Parkinson disease against so-called Parkinson plus syndrome. Parkinson plus syndrome at the beginning usually have some signs which are not common for typical Parkinson disease. So, for instance, orthostatic hypotension or some non-basal ganglia signs like gaze palsy might be present in an early stage of the Parkinson plus. But because there are several so-called Parkinson plus syndromes, I would probably say the clinically most important differentiator are tremor at rest and asymmetry of the expression of a clinical picture.
Perfect. Thank you, Dr. Babich. Another question, if highly experienced raters are being used for a trial, is it really necessary to have such comprehensive and involved training?
Okay, thank you. Yeah, that's a very good question. It is, because first and foremost, it standardizes approaches to the ratings. Study raters may have a great deal of experience with the UPDRS and other scales, but they could have received training from other trainers, from other studies, and there is a potential for variability with that. So by training for your particular, or for this particular study, it ensures more consistency and a standardized approach to the ratings and the scales.
Perfect, thank you. Okay, another question. Are you running any studies that utilize mobile/digital technologies to find, screen, and enroll patients and gather PRO data during the study?
Yes, we do have experience running studies that utilize ePro and eCOA solutions. Yes.
Okay, perfect. Okay, here's a question for you, Dr. Babich. What are some of the most difficult challenges in diagnosing early PD? For example, differentiating from other diagnoses. And what are some of your recommendations in overcoming those challenges? I think you may still be on mute, Dr. Babich.
Oh, excuse me. Can you repeat the question, please?
Sure. What are some of the most difficult challenges in diagnosing early PD? And some of your recommendations for overcoming the challenges?
Well, basically, it is very important to have a clear medical history of patients. Very often we are neglecting premotor symptoms and signs, and one of the leading symptoms which patient can recognize is a loss of smell, and olfactory problems might be first symptoms. Usually, sometimes the trivial pain, for instance, in the arm, might be a consequence of early bradykinesia on asymmetric type, which many people are basically attributing to arthritis or similar. So I guess the most important part is to have a sufficient time to discuss with patient every particular symptom, which could be attributable to Parkinson's disease in the early stage. Of course, very often you cannot make a diagnosis at the first visit, so you need probably to observe patients for a few months to see the progressing of some symptoms, or sometimes if you have a clear picture of the motor Parkinson's disease expression, treatment with the levodopa, initial treatment with levodopa, and very good response is probably one of the best biomarker in early diagnosis of Parkinson's disease. Of course, if this treatment should be requested, usually patients should have sufficient symptoms to be treated, which is usually in the UPDRS motor part, something like 10 or more points. So I think this is the most important in the early stage.
Thank you. And we're 2 minutes away from the top of the hour, so we'll do one more question before we wrap things up. The question is, how many Parkinson's disease trials are worldwide currently working on, and with how many sponsors?
We have 3 trials currently ongoing. 2 have completed recruitment, and 1 is in startup.
Perfect, thank you. Okay, thank you, everybody. With that, I'm going to wrap things up. As a reminder, all attendees will receive a link to view the on-demand recording. That email will come to you most likely tomorrow. If you did happen to ask a question that we did not get to, we will reach out to you individually. Please note that there are some handouts available for you to download in your control panel. So if you would like to learn a little bit more about our expertise in Parkinson's, you can find those handouts in your control panel. Thank you again for joining. We hope you found our webinar informative, and have a wonderful day.
Thank you.